Okay, good morning, and I hope you all came home all right yesterday and that your muscle ache is not too bad. So this is session 5 on methodological advancement in bioenergetics. And Christiane couldn't be here, so we will start with Erich.
Well, I'm Christiane Ceccato. I'm sorry that my voice has changed. So Christiane is our specialist in calcium. I'm not. But since she cannot give the talk, we have substitution strategies. So I'll try to give you a very, very simple message. Mitochondrial uptake capacity is not a term that is generally used, but we are more familiar with the term calcium retention capacity. And as terminology freaks, we try to convince you that there is a very, very important difference between the two. Studied in isolated mitochondria, not only in controls, but also in the presence of cyclosporin A. Now, how do I move to the next slide? Not just click. So I have still 10 minutes. Okay, I don't think I have to give to this audience any introduction about calcium uptake pathways. You see, just to get you warming up, a little figure here. And of course, we have the VDAC, we have MCU, we have delta psi as important triggers for the mechanism of calcium uptake. We have the MCU complex, which may open up under specific conditions. And of course, we know all about the importance that calcium triggers many, many factors that are needed. There is the calcium retention triggered, or calcium uptake triggered calcium release, permeability pore. Well, we are, I think, quite familiar with this, but what is the difference between uptake and retention? So we usually think of a very simple model when measuring calcium retention capacity. Here in this lower slide is the calcium concentration. We titrate calcium, say in isolated mitochondria, and calcium spikes initially and is taken up. And as we go through cycles of calcium titrations, then we reach a point of permeability transition where calcium release is triggered. And this point is the calcium, marks the calcium retention capacity. But there may be incomplete uptake. And for that, it's not just enough to titrate calcium and sum up the titrations, but we have to measure calcium in the medium. And this is now our important message, that the calcium uptake capacity may deviate very much from the retention capacity, because if there is accumulation of calcium in the medium, incomplete uptake, then of course what is taken up, Christiane would say what is uptaken, with her Brazilian approach. So this is the incomplete uptake, and we have to subtract the calcium accumulation in the medium. And therefore, the calcium, the difference here will be, in terms of uptake capacity, very much be dependent on the mitochondrial concentration in the respiration chamber. So now we combine respiration and calcium measurements to evaluate the modification of respiration and the calcium uptake versus calcium retention capacity. The studies with isolated mouse liver mitochondria, we have, of course, a specific respiration medium, because you cannot take just the MIRO5 for measuring calcium uptake with the EGTA concentration, so you see the modifications here. Calcium chloride titrations were performed in 5 micromolar steps, and we have added 1 micromolar cyclosporin A for the experimental group. And the left part, calcium green, is of course our sensor for fluorometric measurement of calcium concentration. Now the experiment goes like this. You have time, and you see this is 1.5 hours. You have in the control respiration with isolated mitochondria, calcium green, glutamate malate, ADP, oligomycin, and EGTA added initially because we never have 0 calcium in the medium, so we have to have some EGTA. And now these are the 5 micromolar step titrations. Respiration in the leak state now is slightly increased as we go through calcium titration cycles. And in the control, then you see here the inhibition of respiration when calcium concentration, no, this is not the concentration, this is the signal, just the raw signal of calcium green. And here you see when respiration is becoming inhibited, then you have the permeability transition. In the control, and much, much higher uptake or retention capacity in the presence of cyclosporin A. So there are 2 challenges. One challenge is the calcium green calibration is not just a linear, simple thing, and therefore just read through these equations. I will not explain them. You get the message. There is some technical aspect of calibration. And you see in the trace here, there is a big, big increase in calcium concentration in the medium, and the accumulation of the actual uptake rather than just the titrations, that is now giving us the difference up to this stage where calcium release is triggered. You see already there is a difference between uptake and just the sum of all the titrations. And the data are this. So if we just give the classical calcium retention capacities, this is the controls, 300 nanomole calcium per milligram mitochondrial protein. This is in the presence of cyclosporin A, and that's the difference. So that will be the classical calcium retention capacity message. And if we compare now the actual uptake capacity, that's the difference. This is the uptake. This is the retention capacity. In the presence of cyclosporin A, even percentage-wise similar, but more dramatic. And the difference between the two cancels, sort of. That is the simple message that we do need to distinguish uptake versus retention. And then Christiane looked also at the effect of our fluorophore, calcium green. We know fluorophores may, say, amplex red may influence respiration, or particularly the cations that we are using, safranin or TMRM. They influence all respiration. With these protocols, calcium green does not, at the concentrations applied, influence respiration at all. So, conclusions, calcium, now, as we saw yesterday, everybody was here looking at the timer. We were here all to get our present. So I'm here to get the present. I have 2 minutes and 20 seconds to get to the conclusion. Calcium did not inhibit respiration. Calcium retention capacity was dramatically lower than calcium uptake capacity. No, that's the wrong thing. Christiane, go back to calcium uptake capacity was lower than calcium retention capacity in mouse liver isolated mitochondria, and the calcium retention capacity dominates in the literature, and this needs to be taken care of when we compare liver, kidney, various sources of mitochondria in terms of calcium uptake capacity. I think that is the physiologically important thing, not the retention capacity. I thank you and my collaborators for taking part in this session.
Thank you, Erich. Any questions?
Nice job, Eric. I know that can be difficult sometimes, especially when you have very little notice, I expect, to stand in. I just have a question about, the old-fashioned way to measure mitochondrial calcium tolerance and response was just to look at swelling. Remember the intrinsic absorbance, right?
Same thing. Retention capacity up to swelling and then...
Right.
Yeah.
Can, with the next-gen O2K spectrophotometer module, can it, assuming we had the right filter, I want to say it's in the 550, 560 range for swelling of the mitochondrial absorbance. Could that be monitored along with, since it's fluorometer?
Yes, and you don't need the next gen for that. I mean, this is not next gen, this is our O2K fluorospirometer, and yes, you can do this in the O2K, you can do this, well, in the next gen you can do everything, but this is also for the O2K, yeah.
So you could have the calcium green signal and a swelling signal at the same time?
At the same time, there may be interference.
Okay.
Yeah. So many of the multi-sensor approaches cannot be combined. We can combine coenzyme Q and safranin, so that can be combined. And the controls, whether you can combine now with the next gen and Q, is all with cyclic voltammetry. So this is the quality control. Cyclic voltammetry diagrams tell you whether you can or not combine. Yeah.
Thanks.
Just a follow-up. So if you're doing replicate samples and they're highly reproducible in terms of your calcium uptake, can you, in a parallel chamber, then do the swelling assay?
Yeah. The approach is of course, you have 2 chambers per O2K, you have many O2Ks, and you can run in parallel. And with respiration always measured in every chamber, that is the connector. So you can really run parallel multi-sensor assays and connect them in terms of reproducibility by looking at the respiration. But you have to be careful if you compare respiration in the presence of safranin and calcium green. Safranin has a dramatic effect on some mitochondria, and it's not always that safranin is the bad guy. It is really mitochondrial specific which cation inhibits more. And it's not only complex 1. So the literature really blames complex 1 very much on the problematic target for cations, for membrane potential. It's not just complex 1, it's also the phosphorylation system. It is sometimes rotenone succinate is bad, and complex 1 linked respiration is better. No predictions, test it.
Okay, we still have about 3 minutes for questions.
Then I get 2 presents, right? We can give the time to the next speaker.
Thank you.
Thank you very much.
So next speaker is Julian Serna from Brazil.
Hello. To change the presentation, what I use my hand. I don't have problem. It's working. And sorry. And if I change the things here, it works with this.
No, just this.
Does this one. So, hello. Thank you for the introduction. invitation. So today I will share with you a methodology that we developed in the laboratory to measure mitochondrial calcium efflux in isolated mitochondria and also in permeabilized cells. So why calcium? Well, calcium regulates several aspects of mitochondrial function, and that includes the transport of substrates and also the activity of several dehydrogenases in the mitochondrial matrix. That leads to an increased flux to the metabolic flux in the tricarboxylic acid cycle, which have consequences for ATP production and also for the production of biosynthetic precursors. But calcium overload may also lead to mitochondrial permeability transition. And this phenomenon is characterized by the formation of non-selective and high conductance pathways for the release that leads to the dissipation of the electrochemical gradient, and they together are known as the mitochondrial permeability transition pore. Then it's like imperative to have pathways that allow for the extraction of calcium. And uptake and efflux pathways are independent ones. Uptake is mediated by the MCU complex, or retinal red sensitive pathways, and we have for efflux the calcium proton exchanger, and also we have the NCLX, which mediates the sodium-calcium exchange. And both of these systems depends on the electrochemical gradient of protons. And NCLX recently have emerged as a very interesting proteins. It have shown to be essential for cardiac physiology, and indeed the overexpression of this protein has shown to protect the heart of mouse against damage mediated by ischemia reperfusion. And in our laboratory, we have shown also that caloric restriction upregulates NCLX. And well, maybe that's related with the cytoprotective effects induced by caloric restriction. So, but protein levels does not mean protein activity. So we were like trying to measure calcium efflux, the activity, but there is no consensus in the literature for measuring calcium efflux. So here we develop a method, and I will describe now. So we use calcium green 5M. This is our extra mitochondrial calcium probe. It also has a low affinity for calcium. And we measure the changes in the fluorescence of this molecule using a fluorimeter. So here is like a typical calcium efflux trace. So the first part for measure calcium efflux is to put calcium inside the mitochondria. So then you can measure how fast mitochondria release that calcium. As uptake and efflux can take place at the same time, and uptake may mask calcium efflux, you need to inhibit calcium efflux using uptake using retinal red. And after that, you begin to see a small increase, a slow rate release of calcium, and you can stimulate it, for example, using sodium. And finally, we have the calibration step that is essential, and you have to perform this in each trace. And what we do here is transfer the fluorescence value in calcium concentration values. So here we describe some critical points that are essential for reliable measurements of calcium efflux. And the first one is related to calcium upload. And it's very important to avoid mitochondrial permeability transition induction. So as it was previously commented, related with the calcium retention capacity. So it's like the maximum amount of calcium that can be uptaken with a mitochondria before the generalized mitochondrial permeability transition induction that we can see here because mitochondria begin to release calcium after they have been challenged with different calcium additions in the medium. So, but the point is that not only here we are observing mitochondrial permeability transition, we can observe permeability transition in very early phases during the trace. And it's a simple way to check this. And what we did is that we put different amount of calcium in the mitochondria. All of them were under the calcium retention capacity, and we put retinal red after the calcium upload. So as you can see here, increasing the calcium load increase the rate of calcium efflux. But what is interesting is that all that can be inhibited by cyclosporin A, demonstrated that is permeability transition. And here is a quantification of what we observe. And this is not something new. It was previously shown that permeability transition is a populational phenomenon. But we need to keep this in mind when measuring calcium efflux, because I can use like any concentration above calcium retention capacity. I have to use one in which I do not induce mitochondrial permeability transition. I think it's like not working. It stops to work. Sorry. That's right now. So, sorry for the... So we need to avoid mitochondrial permeability transition induction. And why? Well, first, as I said previously, both calcium-protein exchange and NCLX activity depends on the electrochemical gradient of protons. And second, because mitochondrial permeability transition pores exhibit a high conductance, which will mask calcium efflux through the other pathways that exhibit a lower conductance. So secondly, it's very important to calibrate the traces, and this because it's very commonly to observe in the literature that people compare changes in fluorescence, the rate of fluorescence change, and that is not accurate because that is assuming that the constant dissociation is equal in all that conditions. And we have seen, for example, when you change the ionic strength of the solution, if you are different composite solution, that dissociation constant change. So it's essential to determine that parameter to calibrate our traces. And finally, titrate everything. People use like different concentrations, and some are like very high. And even when you have a selective inhibitor, you may induce some undesirable effects. We have observed a CGP, for example, may induce mitochondrial permeability transition. And also ruthenium red is able to inhibit sodium-stimulated calcium efflux. So having all this in mind, we try to solve a problem. And is the existence and the functionality of NCLX in their liver mitochondria. And it has to say for some ones that it exists, for others that it doesn't exist. So we try to assess this. So we isolate mitochondria from rat liver for differential centrifugation. And you can see here the typical traces. You have the basal one in the absence of sodium. We were able to measure sodium-stimulated calcium efflux, and also to characterize the function of this protein, we measured calcium efflux in the presence of lithium. Lithium NCLX different from neutral sodium calcium exchangers. It's able to exchange calcium with lithium. And finally, we were able to inhibit this with a good inhibitor for NCLX that is CGP. And also this was not an artifact of addition because potassium that is not expected to induce NCLX activity was not able, and well, we don't see any difference. We also see the same behavior using mouse liver mitochondria. So NCLX is present and is functional in liver mitochondria. And then we move to a more physiological system. This is permeabilized cells. And this is a good approach when you have like a small samples you have small samples. And for example, for the ones that have tried to isolate mitochondrial cells, it's very difficult because you need a lot of cells. And well, here we first permeabilize cells. Here you can see the digital infiltration. And we also use a control for the other mitochondrial permeability integrity. And after this, we use the same protocol that we established for isolated mitochondria. And it works pretty well. We can see here the basal calcium efflux. We can see the sodium-stimulated calcium efflux. And we see that this can be inhibited by CGP. So demonstrated that it's NCLX. So in conclusion, we describe a method to measure sodium-sensitive and insensitive calcium efflux in isolated mitochondrial permeabilized cells. We demonstrate that this is very important to avoid mitochondrial permeability transition, and also that NCLX exists and is active in the liver mitochondria from rats and mouse, and also in permeabilized liver cells. And finally, I would like to thank you all these amazing people with I work, and thank you.
Thank you, Julian. And Julian actually had 20 seconds in his advance, so he was fine on time. Any questions? Yeah, Eric?
Thank you very much for this great talk. Did you observe in the presence of sodium differences in the calcium concentration that was increasing? In my previous talk with the upscaling or increase of calcium in the medium, you showed exactly the same. So did you see that there were differences in the presence of sodium, lithium, etc. in your different protocols?
I didn't understand what you
Okay, so you presented different protocols with sodium sensitive, sodium insensitive, calcium retention capacity. But did you see under these different conditions that the calcium that increases as you titrate calcium, that these concentrations in the medium are dependent before you reach permeability transition on sodium, lithium, potassium?
Okay, I think, we measured sodium stimulated calcium efflux with different calcium loadings in the mitochondria. And you can observe when you increase calcium loadings in the mitochondria, you can, in the presence of cyclosporine A, for example, as you increase the concentration of calcium, you put sodium, you get higher rates also. But you need to add other things like ATP or cyclosporine A in the medium. But without that, if you put a lot of calcium, when you try to modulate with sodium the calcium extraction, you have a very high slope of calcium release. And when you put sodium, actually you have a lower slope of calcium release. I think it's that you were saying.
Thanks, Julian. It's important to have these kinds of methodology papers out there to follow. I know for experienced and obviously new investigators. Another thing, and we may hear about this from Nicola later in the session, but obviously the quality of mitochondrial isolation could be a major effector of how consistent your data is going to look. Did you notice this in your repeats? I mean, we saw some nice data, but when you're doing this day to day, do you see a lot of variation depending on the quality of whether the animals are fasted, whether the liver isolation, mito isolation protocol went well? How much variability do you see?
That's a very nice question. So it was very reproducible. We have seen very similar experiments. All the animals that we use are feeded. Actually, there is a work from a friend of us, where he shows that when you have people usually fast animals when they are going to isolate mitochondria from liver. But what he saw is that when you don't fast the animals, they are feeded, you have like very better coupling, higher calcium retention capacities in that. And a second thing that I would like to say to that is that we saw difference using different strains of mice. In that we have the Jackson animal, the C57 from Jackson, and because they have a mutation in the NNT, the nucleotide transhydrogenase, they have lower calcium retention capacities. It was very difficult to measure calcium efflux stimulated by sodium in that animal. Actually, for that was the reason why you use the TAC animals.
Great, thank you.
And have you looked at differences in like anesthesia? How you?
No, we didn't try that, but we used ketamine, chilacin, and aspirin. But that's interesting because CGP, for example, is an inhibitor, is the benzodiazepine. And I think.
We have about 1 minute for one more question. Otherwise, we thank you very much. And welcome next speaker. It's Doris Wilflingseder from Austria.
So, good morning, and first I want to thank Erich for the invitation. Also, I'm not really into mitochondrial pathways and respiratory investigations, but what we are interested in is how to optimize respiratory models for SARS-CoV-2 research, but not only, of course, for SARS-CoV-2 research. So when SARS-CoV-2 popped up, we already had an optimized respiratory model at hand because we were interested in fungal interactions of respiratory and lung cells so at the barrier sites. So therefore, we already had an immunocompetent model at hand when we got the first patient isolates from SARS-CoV-2 from the internal medicine. And therefore, we could start immediately when we had those patient isolates expanded. And first, we had some looks at interactions of SARS-CoV-2 just with the epithelial cells, but then we also tried antiviral sprays and drugs. So here I want to introduce the models we are using. The first model we are using is the air-liquid interface model, which I will explain in a minute. But here we just did several optimizations to end up with a very well differentiated pseudostratified epithelium. And within this model, we can measure several readout parameters, such as transepithelial electrical resistance for epithelial integrity. We have lots of imaging techniques to look at the interactions of the pathogens at the epithelial barrier sites. And from the basolateral subnatant, we can measure cytokines, we can measure anaphylatoxins, but of course we can also measure viral load and infectivity of the viruses by adding the subnatant on SARS-CoV-2 susceptible cells. And of course, we can also do measurements from the apical site, which is the mucus-producing site at the airface, because here we can also measure the viral load, for example, to see which viruses are secreted on the apical side or on the basolateral side. This is a very physiologic system because we can also add immune components such as dendritic cells, macrophages, T and P cells, but also, of course, complement or other soluble factors of the immune system. But, of course, we can't do any high-throughput analysis. But I will show you a little bit later what we are using for this one. So how did we optimize the system? We wanted to go really animal-free. And normally, within the transfer system, red tail collagen is the standard scaffold for seeding the cells. So we found a substitute for the red tail collagen, and now we are using the birch-based cellulose Grodex. But of course, we first had to compare the gold standard with the novel method. And the second optimization we did in this system was to seed the cells not inside the transfer, but on the bottom of the transfer. Because by this method, you can really use the same cells for multiple analysis. So for live cell analysis, you just have to take the transfer into a glass bottom plate and can do live cell analysis over time. And you don't have to sacrifice for each and every analysis a transfer. And this is very nice if you want to monitor the differentiation of cells, if you want to monitor, for example, multiple exposures in one specific transfer. And these were the optimizations we did with the air-liquid interface model. And this is how it looks. So we have a nicely differentiated pseudostratified epithelium with ciliated cells, which, how can I switch on the video? Sorry. One moment, please. One moment, please. Yeah. So where we, here you can see nicely differentiated ciliated cells. Sorry that it does not really work. But the differentiation was very high in this system. And of course, we also had mucus-producing cells. Can we go back to the presentation, please? We also had nicely differentiated. Here we go. We had nicely differentiated mucus-producing cells, here shown in orange. And by directly comparing the growth in Grodex, in the cellulose-based Grodex matrix versus the red tail collagen, we found that we had a faster differentiation of the cells for about 5 days. So the cells grown in the red tail collagen looked like this after about 5 days. And this is how the cells look like. And here we also stained for the mitochondria to look at the viability of the cells. And they feel really happy in this system. Here you see the ciliated brush border, and you can see the nucleus stained in blue. In the next step, we also applied perfusion, because this is what our cells are exposed to in the body, and it mimics more likely the in vivo conditions. And also under perfused conditions, we found a faster differentiation of the respiratory cells in the air-liquid interface compared to static conditions, which was also shown when looking at the cilia. Here we see lots of immature cilia, while under perfused conditions we saw really a high differentiation of the cells. The other system we are using are lung organoids, and as most of you will know, the basolateral side is the side which is looking to the medium in the organoids, while the apical side is in the center. Here we stained for the 2 receptors of SARS-CoV-2, ACE2 and TMPRSS in violet and in green, and as you can see, it's in the center. And that's not how we want to infect the cells. Therefore, we repolarized the tissues and now are working with epicloud organoids, where you have the cilia on the outside. And thereby we can easily infect those organoids and also add, of course, drugs to the organoids. And that's the system we are using for high content analysis. And whenever we see that a drug is working, we go to the more physiologic system, where we can assess also at immune cells. And fortunately, all those experiments really worked out nicely. And we had really nice publications over the last time. And I just want to show you some pictures from those publications. So here we infected the air-liquid interface model with SARS-CoV-2, and of course always compared it to uninfected cells. Here you can see the nice intact epithelial layer with mucus-producing cells in orange, with cilia in green, while in the infected cultures you can see big holes which is caused by tissue destruction. You can see virus-infected cells as well as virus particles, and what you can immediately see is a mucus hypersecretion as well as mucus plug formation. And what was interesting is that this is also seen in critically ill COVID-19 patients. Another thing we did was to use an antiviral spray, which is commercially available. We treated the tissues and then infected the tissues, and we saw that the spray completely rescued the tissues from the damage caused by the virus, which is also shown here. Here, with applying the antiviral spray, we could not see any activation of the innate immune system, and we could not detect any tissue damage, which is also shown here by this increasing the TARE values again. So with this, I want to thank you for your attention. We are always also combining our in vitro data with data from patients, and we For this, I also got the Austrian State Prize for the promotion of alternative needs. Of course, this is not a solo project, but also performed in a team, and I want to thank all people involved in the system. And I also want to thank you for your attention.
Thank you very much, Doris. Any questions?
Thanks, Doris. That was a impressive model, beautiful images. I'm curious. I love these 3D models, multiple cell types, and as you know, the challenge is how long do they retain that phenotype and differentiation structure in culture over time? So I'm curious, how long can you maintain them? How long do they last like this?
Yeah, thanks for the question. So for the air-liquid interface model, we just kept the cells in culture and monitored it over time, and we could retain it over 2 years. So we did not have a really detailed characterization, because then we would have to really have huge amounts of transfers. But we did live cell staining with just a nuclear stain and mitochondrial stain, and they are also a stain for the cilia, because this can be done by live staining, and they were really nice for 2 years.
That's impressive.
Of course, not the organoids. The organoids we are having in culture for about 2 to 3 weeks, but
Great.
that's okay.
Thanks.
You're welcome.
Great talk, thank you. I have a question related to how these types of methods or other methods may be applied to study post-acute COVID-19, or sort of the sequela of symptoms now that we see that are sort of the large push to sort of figure out how we understand the sort of trajectory of a patient after acute infection.
Yeah. I mean, I think because what we saw is that when we infected the cells at a multiplicity of infection, which is about the same as we are exposed to, then the cells are they show holes and they are dying after 5 to 7 days. But I think maybe if we expose it to much lower concentration, then we would get kind of these chronic symptoms. But and we also saw differences with respect to the variants of concern, because, for example, with Omicron, we saw that it did not hurt the cells so much as Delta, for example, and but I think you would just have to really go down with the concentration of the virus to get some kind of chronic exposure, or maybe also at really low concentrations several times, but we have not tried that yet.
Question here. Yeah, thank you very much for your presentation. I was wondering whether your cell model is also applicable to other cell types. How easy would that be to implement? Do you know of any groups that you work with that have tried this model?
I think that I mean, these models are also common for intestinal cells or also other epithelial cell layers. So I think, I mean, as said, we optimized it for lung cells, but I think it's not so difficult to really apply it to other cell types too.
Okay, great, thanks.
So I think we can move on to the next. Thank you very much.
Thanks.
All right, so for our next presenter, we'll have Martin Strehle, speaking in place of Peter Wolf. And it'll be physiological metabolic RTCA on automated platform for short and long-term applications.
Okay, so
the videos won't play.
so it's a video.
Just like So, hello everybody. Thank you very much for the invitation, Erich. Ladies and gentlemen, welcome and thanks for having me. My name is Martin, and today I'm talking on behalf of Peter, who is unfortunately not with us today. And I'm just giving you a short introduction of what we understand as a physiometabolic real-time cell analysis. Technology, yeah. So, we at Zeiten are a technology company, so therefore I should understand how to use the presenter. And we are measuring the whole living cells in culture, and that is what we are doing, not presenting every time. So every cells, we measure the input signals from its cellular environment, and we measure for nutrition, oxygen, drugs, or physical stress. And we are reacting, and cells are reacting to these input signals in a general matter with like parameters changing in oxygen consumption, in extracellular acidification, and morphology or proliferation. So, and this is where we are looking at. But to understand how we are doing this, we just go into the system of life. Cells are reacting to the environmental changes, this is what is the output. And when we control the environment within an atmospheric chamber, and also we add a media fluidic system, which exchanges media and nutrients, and we all measure the changes in the parameters with multiple sensors in real time, we get a system like what we have developed, and we put everything what we have in control, actuators and controllers, as well as sensors in one device, which is then the CyRIS-Flox system, which is able to perform the automated cell-based assays in real time and allows dynamic evaluations of multiple parameters simultaneously. So the technology in detail is that it's built within an atmospheric chamber where we have an incubator, and we built quite a lot of technology inside. And we control their temperature, humidity, and the oxygen content from 1% to 21%, which is normoxia, depending on the ABC of hypoxia. But inside the chamber, there is a robotic system that supplies media and drugs to our living cells in our special sensor well plate. And this sensor well plate is more or less the core of our measurement system, and it has 24 chambers, and each is equipped with a microfluidic channel for media supply and the sensors for oxygen and pH, and also for impedance, and with a dedicated window for allowing us for the microscopy. And you just see here the media inlet, the microfluidic channels, and then you have the micro-reaction chamber where the cells are added, and with the pipetting robot, we remove media from this side and we add media, nutritions, or any drug candidate on the other side. Under the sensor plate is a movable digital microscope, which continuously reads the optical sensors and takes images of the cells. So which data can we produce with a system like this? So the first example I want to show you is a hepatotoxic assay with liver cells treated with 4 different concentrations of APAP, the drug in paracetamol. So we started with 50,000 cells and investigated them over 48 hours with 1, 5, 10, and 50 millimolar APAP. We made a 12-hour pre-measurement time frame and the baseline, and then we made a 24-hour treatment, and at the end we made a 12-hour washout. So I could talk about these graphs alone 20 minutes because there is already out of this one experiment a lot of data. But as a quick description, what you just can slightly see is that within the oxygen consumption rate and within the extracellular acidification rate and within the impedance data, you just get a dose-dependent reaction in all measured parameters, and the cells are able to recover after a washout up to a concentration of around 10 millimolar. And this data can be used then to further downstream calculations, like example here, the EC50 of APAP only in the oxygen consumption rate and at different time points, and therefore you just can create such things like a dynamic EC50 over time. and also about different therapeutic windows where you may expect changes in the EC50. So, since we're also doing microscopic images of these experiments, we see the different concentrations over the time, and red is during the treatment, and the cells show normal behavior up to 5 millimolar, and under the treatment of 10 millimolar APEP, the cells do not really proliferate anymore. And they already show intracellular accumulations, but proliferate again after the washout. And cells with 50 millimolar APEP die very quickly and crumble together. So we have here OCA, ECA, impedance, and imaging all together showing the story from different point of views already from one experiment. And since now we have learned that long-term experiments can go up to 2 years, mostly if the cells survive, but we also did long-term experiments. But our long-term is not very defined, so we have, in our meaning, experiments that are longer than 3 days. And here we measured the same cells, liver cells, with over 6 days, and we used 30,000 cells, and we used the concentrations 5 and 10 millimolar APEP respectively. And we made a treatment of 6 hours washout, followed by 42 hours treatment. And what you can see here is that the oxygen consumption rate, it's quite challenging, because already in the control, it's growing over a wide range of 50 to 800% in this normalization, and it goes into a plateau phase, quite at the end of the experiment. So there is room for protocol improvement, but the important thing here is that there are different reactions of the cells under treatment. And this is what you just can see with 5 millimolar and 10 millimolar respectively. So these cells are showing recovery effects, but then are also showing the possibility or the unavoidable point of death, whereas the 10 millimolar after this treatment already is dead. And because we take a picture every 20 minutes, we make the repositioning of the microscope, and since this is very stable, we can make connected movies, and it would be good if you just can start the movie, because what you just can see, it's like making both, I'm sorry, simultaneously at the same time, but maybe I just need to click it like this. So it's my fault, sorry. So what you just can see is like the movies of this 5 millimolar and 10 millimolar at the same time, and with the oxygen consumption in the bottom, you see these images are taken at every time point, and also then you have the respective graphs, what is going on within the oxygen consumption rate. And of course, what you see is like in the graph that you see during treatment, there is less proliferation and already like this integration of the cells, and then the 5 millimolar, they are already also decreasing the viability at the end of the experiment. And I think just closing this, well, nearly all of you are familiar with the mitochondrial stress test, and this is just something that we also can do. And we're already also doing protocols where we automate the whole process so that we have like an ongoing pre-treatment or pre-experiment, and then afterwards we have an automated mitochondrial stress test performed in the platform. So that means that you do not have to touch your cells anymore. For example, we did a macrophage polarization experiment, and afterwards an automated mitochondrial stress test where you just can receive all the relevant parameters for respiration investigations. And coming to an end, we have developed this platform, and we would like to show you even more possibilities, but we are eager to develop more possibilities, what we can do with this platform together with you as the scientific community. And I think we have forgot already some things during the development, so we are happy that you just come by to our booth or with your project idea or with any idea for collaboration, and well, we are happy to talk with you. Thank you very much.
Great presentation. It's open for questions.
Very interesting talk. I was wondering whether the respiratory capacity under this system is different from the O2K or the Seahorse system.
Well, that's a good question, and I think Erich invited us, since we know each other side quite some years, but we are up to make these investigations what is possibly synergistic in performing experiments with a Cyrus, with an oxygraph, and then also how can we get the connection of what differentiates us to the seahorse experiment. Because from our side as well, we not only make the snapshots on a short stage for mitochondrial assays or mitochondrial stress tests, but we are also looking for these long-term effects and maybe for a locked-in state for the cells, and then like the effects on oxygen consumption or on other morphological behaviors from the cells.
I have a quick question over here. Can you also measure like H2O2 production in vivo, especially with your imaging, because that might be interesting?
Well, we have not yet tried to, because, well, we had not chance yet to do so. What we have measured is like if the adding of H2O2 on our sensors has some negative effect, and this was not shown. So we are like positive to measure something, but, well, it depends on the research question.
Eric?
Thank you for this presentation. I suggest that you should differentiate yourself in terms of terminology from the seahorse world, because to say ATP production for something which is a difference in oxygen consumption is just not science. But it does not relate to the measurement. It relates to the presentation of the measurements, and we should be scientific and not use misleading jargon.
Yeah, thank you very much.
Adam?
Hi, Martin. Yeah, beautiful system. I might have missed this, so my apologies if I did, but the pH and the O2 sensors, they're underneath the well, right? Are they sampling media that's removed from, micro samples that are coming from the well, or are they actually kind of in the well measuring the monolayer from below, or,
Yeah, good question. And, well, it's indicated in the, image, I think. Where do I get it? Technology. So that's, I think, what the question was is that if these sensors are under the well and that we have like a media layer between the cells and the sensors, and that is not the case. So the sensors are within the cells and cell environment within the microreaction chamber. So that means that we have the sensor spots in every well bottom where the cells are then growing around and are measuring the microenvironment within this microreaction chamber.
Right. So, and we've done a little of this we were using single cells oocytes, and the positioning of the cell was important, or at least I won't say critical, but if it wasn't right there, and you were counting on oxygen diffusion gradients throughout the well with single cells, that's a big problem. In here, I think you said about 30,000 cells. Are they confluently plated so that you've got the whole surface covered? How important is that, or if you have patches? Could that throw off your measurement because it might not be sitting right in the same spot? You know what I'm saying?
Well, this is actually also a good question, because what we have measured is like when we make this pipetting system and we have this unilaminar flow within the microfluidic channels, we generate this kind of movement within this microreaction chamber so that you have the settlement of the cells more or less equally distributed. And therefore, we are at the moment fixed to adherent cells because when we use suspension cells, these cells are washed away. And this is also what we just then can see with the impedance measurement, that we see there is this ongoing settlement of the cells, and with the microscope we just can control if they are evenly distributed.
Great, thanks.
Yeah, you're welcome.
In the back?
Okay. Hi, over here. Did you try to measure at a different atmospheric oxygen concentration? You mentioned you did it at 1% oxygen, and you said that was normoxia. I agree with that in the short term. If you incubate your cells for 24 hours at 1%, you can see differences in gene expression, at least for glycolysis, oxidative phosphorylation, cholesterol synthesis, whatever. So the question again is, have you tried to measure at a different oxygen concentration?
Well, we are right at the beginning of going, I think, below 5%. At the moment, we just did experiments with around 5% oxygen concentration within the device, just to have like a kind of a control of what still is possible, because the oxygen consumption needs to be there, and the cells need to have something to just live on. Otherwise, well, we have problems. This 1 to 21% is what the device is capable of. So we cannot go by the device control below 1%. Yeah.
All right, with that, we'll move on to the next presentation. Great job. So next we have Rupert Ecker, contextual tissue cytometry with artificial intelligence, single cell analysis in situ.
Chair, Erich, ladies and gentlemen, good morning. Over the next 10 minutes, we will leave the core topic of this conference, as I'm going to present you a technology platform that allows us to better understand cells in their native environment in situ, so mainly in tissue. This technology platform is made commercially available through my company, Tissue Gnostics, but it also relates to my work as adjunct professor at Queensland University of Technology in Brisbane. What is tissue cytometry? I assume you're all familiar with flow cytometry, a technology that can measure antibody-mediated staining in single cells with one major limitation. These cells have to be in suspension, as the name flow cytometry already suggests. Tissue cytometry brings this idea to the slide, to the microwell plate, adherent cell culture sections, adherent cell cultures and tissue sections. You can see here we can automatically identify individual cells in tissue context and identify subpopulations. Of course, we can also look on multicellular structures like tumor areas, glands, glomeruli, vessels, epidermis, whatever, and then do what is called spatial immunophenotyping, so measure single cell populations in relation to histological context. We can also look into subcellular compartments and measure structures like cytoskeleton, mitochondria, or also pathogens like bacteria, viruses, or intracellular parasites. Very quickly, the company Tissue Gnostics is located in the city of Vienna in Austria. At least the global headquarters is there. We have an international structure and network of subsidiaries and offices. The company has been founded in April 2003, so next year we will celebrate our 20th anniversary, and I would like to emphasize that we are manufacturer of medical devices according to ISO 13485. And very important, this month we will actually reach 2,100 reference papers from research groups in more than 60 countries on 5 continents. It's not a single instrument, it's a technology platform based on a high-end microscope for bright field imaging, fluorescence imaging, whole slide confocal imaging, an inverted system where you can work with well plates, do time kinetic experiments or even live cell imaging, high throughput scanning with a slide loader, or a system for spectral unmixing using a spectral camera with a liquid crystal tunable filter. But what makes Tissue Gnostics is the analysis software, and these are our applications. I don't have the time to go into details, but we have a booth outside, and if you're interested, just please visit us. So we see here an example of a colon section in immunohistochemical staining with peroxidase in brown. And the obvious question is how many cells are proliferating, or in other words, how many blue nuclei are brown. We made a survey among more than 200 scientists, and the surprising outcome was the estimations ranged from 1 to 40%, which impressively shows the main problem that is always associated with visual evaluation, that you get a wide range of estimations rather than a reproducible measurement. With our TissueFox platform, we can identify the single cells using the nucleus as a master channel. The yellow contours show you here what the system has identified as single cell, and then measure how much blue and how much brown or any other color, and it also works in fluorescence, is associated with each of the nuclei. And what we get is this flow cytometry-like diagram, or people in flow cytometry call it a dot plot, just that each dot that you see here represents a single cell in a tissue context. We see here the mean intensity of hematoxylin and here of Ki67. Here's the cutoff, very important, based on a negative control that determines the specificity of our staining, and this is our positive population. And we get a reproducible measurement and independent of when and where we do the analysis. One of the features that our clients appreciate most is the ability that you can track back and forth individual cells between the virtual slide and data space. So you click on a cell and it starts blinking in the scattergram, and so you know exactly where this cell is located. This we call the forward connection from image to data. It also works the other way around, from data space back to the image, and then we call it backward connection or reverse gating. You can make a gate like in flow cytometry, but the cells which are part of this gate will be highlighted. So you have an immediate relation between a cellular phenotype and histology. Where in the tissue section are these slides? We also offer extensive machine learning module where you can do a classification of tissue. You define your classes that are relevant in your sample, and then the system will find this on your entire sample or a set of samples. And you can combine this classification with the single cell analysis, and you see here for all the classes that were identified by machine learning, you get also the single cell measurement, and when you have a proper staining protocol, you will know how many cells of which phenotype are where in the tissue. In Queensland University of Technology, we have defined a project that we call the Virtual Histopathologist for Precision Cancer Diagnostics, where we make an AI-based decision support system for histology and use prostate cancer as a model disease. We're working with international partners on this. The project consists of 5 sub-projects. First is the definition of markers that are specific and clinically relevant for prostate cancer diagnosis. This is not really our work, but is more a literature study where we use what is already known. This is headed by my colleague Jyotsna Patra. She will have a presentation this afternoon. The second part is development of antibody kits using a linker technology that allows easy multiplexing staining, so restaining of the same sample. Then in project 3, we combine protein markers with genetic information like DNA and RNA, and then comes the actual AI work, which includes data mining. So when you stain, and this is the main idea of multiplexing, multiple markers, and with multiple I mean dozens, it becomes very difficult to really understand what the data show us. You cannot just make dot plots where always 2 markers are plotted against each other, because you will end up with thousands or even 10,000 of scattergrams, and what does this tell us? So we have implemented also a data mining functionality which automatically finds correlations between markers. So these are the markers, we can hardly read them for whatever reason, but the lines that you see here, the color and the thickness and where the lines go indicate which marker is correlated in which way, negative, positive, or not at all, with all the other markers. And in particular at QUT, we focus, because this is a lot of deep learning work, on what happens in a deep neural network. In a DNN, we usually define the input and see what comes out, but we do not really understand what happens inside. So this is really a purely academic work of better understanding how artificial intelligence can be implied, and this is specifically with relation to regulatory authorities like FDA, for example, where we need to prove that something and show why something works and not only that it works. Bringing all this together, we do what we call contextual tissue cytometry. Here we see identification of single cells of the tumor area, and then we combine these measure masks and can ask detailed questions like here, looking for CD4 positive T helper cells in the context of tumor. These are colorectal cancer. Here on the y-axis is the distance in micrometer. Each cell that you see here is a CD4 positive cell. And now we can see in red all those CD4 positive cells which are just around the tumor, shown in orange, but not further away than 25 micrometer. And we can do this for all the distance zones, and that gives us a very detailed analysis on the single cell level. And it's a measurement, not an estimation. I would like to refer to a review article that has been published by my group last year, where we also focus on this topic. And finally, I would like to acknowledge our partners from the Marie Curie Innovative Training Networks. Tissue has been member in 5 such innovative training networks, and in particular, my partners from the Virtual Histopathology project at QUT. This is a dossier on artificial intelligence, which we have published earlier this year. It's not a scientific article. It is what funding agencies typically would refer to as public outreach activity. Still, it's very informative, as the content comes from our collaborators and partners around the world. And with this, thank you very much for your attention.
Great study and technology and perfect timing. This is open for questions. I guess I can start with one question. So for your tissue cytometry and your tumor kind of analyses, can you also look at mitosocs or other targets?
So when you stain structures, subcellular structures, mitochondria, endoplasmic reticulum, whatever, then the image cytometry software that we have will be able to quantify it in cellular context. Tell you, for example, how much in area or in counts is there. If you look on specific markers associated with these structures, how many of the structures are positive, negative, how strong is the expression. We have special algorithms for finding small dot-like structures inside the cell. It's called the dot finder algorithm that gets, for example, applied to fish fluorescence and cytohybridization or RNA-scope samples, but also structures like vessels or mitochondria.
Great.
Very nice presentation. Yeah. Your slide with your hardware went very quick. Can you elaborate a little bit more on the hardware? Is it applicable to every standard microscope that you have?
So what we offer is a complete system, and the core of which is a high-end size microscope. Tissue is OEM partner of Carl Zeiss Germany. It is possible, if you have a motorized size microscope, that we upgrade it by maybe at a motorized stage and a specific camera and our software. So this is possible. The image analysis software is also available standalone. We support not all, but most of the third-party slide scanners on the market, like 3D Histech, Hamamatsu Nanosuma, Akoya, and so on. So you can read the native file formats from these slide scanners and do all the analysis applications that I have shown, and these are only, I showed very few of them.
Yeah, great. I'll see you later, but thanks.
Any more questions?
I invite you to visit us at the booth outside. There we have more time to discuss.
All right,
questions.
so we'll move on to the next speaker, and
Thank
we can
you.
chat at the coffee bar. So our last presentation of this session is by Nicola Lai. He's talking about effective isolation protocols and skeletal muscle mitochondrial subpopulations and their bioenergetic function.
The presenter. This one? So we go back to we don't use the, okay. And where is the pointer? This one? Okay, thank you. Good morning. Well, my talk focuses on the effect of isolation of protocols, isolation protocols for skeletal muscle mitochondria. And in particular, we are interested to look at their effect on bioenergetic function in rat skeletal muscle in presence of type 1 diabetes. So what's the motivation of our work? We have been interested in bioenergetics function in diabetes for quite some times, and after we study the bioenergetic differences in skeletal muscle, subsarcolemma, and interfibrillar mitochondria in a rat model of type 2 diabetes, we read an interesting article by Monaco that shows alteration of mitochondria, bioenergetics, and ultrastructures in skeletal muscle of patients with type 1 diabetes. So then we got interested to know if these bioenergetics differences were observable also in the subpopulation of mitochondria. So then we decided to evaluate the effect of isolation of the whole population and the isolation of subpopulation of mitochondria on the bioenergetic function in control and drug-induced type 1 diabetes rats. Well, first, we look at the mitochondrial content. So we determined the mitochondrial yields. So here I reported, you can see milligram of mitochondrial protein isolated per gram of tissue. And you see in the whole population, the yields are quite comparable between the control and the diabetic rats. If you look at the yields of the subpopulation in the control group, the sum of them, so SSM and IFM, are still quite similar to the values that we observe for the whole population. In contrast, when we look at the yields for SSM and IFM in diabetic rats, there is a trend to be lower in the IFM of the diabetic rats in comparison to the control. So then we look at the respiration rate in the whole population of mitochondria. And on the left panel, I compare the oxidative phosphorylation capacity of control and diabetic rats measured using complex 1, 2, 3, and 4 substrates. So we didn't see any particular differences. And these results were also confirmed looking at the electron transport system capacity reported on the right panel. So then we compare the respiration rate of the whole population of mitochondria with those obtained from subsarcolemma and interfibrillar mitochondria. So these are the results. In this slide, I just reported those obtained with the succinate, but were similar to those obtained with the other substrates. So on the left, let's start to see what we observe on the left. So these are referred to the control group. The respiration rate for the subsarcolemma, so SSM, are significantly lower than the interfibrillar mitochondria and the whole mitochondria. So this has been observed for the oxidative phosphorylation capacity. It has been confirmed also on the electron transport system capacity, indicated with the letters P and E respectively. In contrast, looking on the right panel, and so with the diabetic rats, we observe something different in comparison to the control. If you look at the SSM rates, these are significantly lower in comparison to the interfibrillar mitochondria, and the interfibrillar mitochondria rates were lower in comparison to the whole mitochondrial rates. And this has been observed systematically for both oxidative phosphorylation capacity and electron transport system capacity. So based on these results, we can say that in type 1 diabetic rats, the isolation of subpopulation of mitochondria, and I'm finishing, is detrimental to the isolation of mitochondria and to the bioenergetic, in contrast to the results that we obtained for the protocol that isolated the whole population of mitochondria. So the key question is, why does this happen only in the type 1 diabetic rats? We don't have a conclusive answer yet on this, but it's possible that the presence of autophagic components that has been found in SSM and the IFM could be largely affecting the bioenergetics function of those mitochondria that are isolated using protocol that can distinguish the two populations. I was finished here. So. Thank you.
All right, I think it's time for a coffee break, and we can ask Nicola questions at the coffee break, right? Perfect.